So welcome to the Advanced Microbiome Analysis. Some of you I've saw just 2 days ago, and there's also some new faces as well. So I'll go over a few things that we talked about with the beginner course, and then we'll jump right into content a little bit quicker today. So my name is Morgan Langille. I am a professor at Dalhousie University. I study microbiomes in lots of settings. I tend to study the human microbiome mostly these days, mostly because I'm in the Faculty of Medicine in the Department of Pharmacology, but I have studied other types of microbiomes and soils and things. I have a particular interest in bioinformatics because I've developed some tools in the past, and we also compare a lot of tools in the microbiome world. And we have quite a range of studies. So we, in the past, I've worked on inflammatory bowel disease quite a bit. More recently, we've been looking at sort of cancer and tumor-associated microbes, and also some work with oral microbiome in kids, and also microbiome of frailty in aging people. So if you have a microbiome, I've probably heard about it, and we can chat about it. Also, I run the IMR, which is a sequencing and bioinformatics services out of my lab, which I'm happy to talk to the people that didn't hear about it from before. So if it wasn't clear before, we do have sort of this format where we have a lecture, which is about an hour, right? Feel free to interrupt me at any time. I'm happy about that. More than happy, actually. It gives me a mental break. And that's followed by a lab, obviously, where then you're going to be more hands-on, and that's already sort of prepared ahead of time. And you'll be getting to do that sort of quietly, sort of on your own, with a lot of support in the room from TAs and myself somewhat. I'm not as good on that side. So that's sort of the format. And I think, overview, yes. And then I guess just for an overview of the workshop, today the first module is going to be really talking about assigning taxonomy to metagenomic data, and also just making sure we're all on the same page of what I mean about metagenomic data. There'll be a bit of an intro to that. And then this afternoon, you're going to get right into MAGs and metagenomically assembled genomes, and that's going to blow your mind, I'm sure. It's very interesting stuff because obviously it's fun to try to extract genomes from metagenomic samples and analyze them. And then tomorrow, so that's going to be led by Ryan. And then tomorrow morning, we're going to get into functions way more. And that's where John's going to lecture, and Tony has a lab supporting that. Right? Yes. Yeah. And then tomorrow afternoon is, I believe, Tony and Robin and John talking about an assortment of things, really. Some functions, some advanced statistics, all that fun stuff. And that's supported by a lab from Tony as well. So it's going to be great. So let's just get into the first one and see how this goes. Sounds good? Any questions before we get started? Wonderful. So I do realize there's just TVs, there's no projectors. So yeah, strain your neck this way and then you can look at me, it's fun. Yeah.
So on the website for the beginner microbiome, there were links to the lecture note, but
yes.
Those are uploaded.
You're not, I'll be there in a moment.
Okay, cool.
Yeah, the reason for that is I borrowed a slide from Robin, and somehow in 3 slides, those take up almost 300 megabytes of space in a PowerPoint, which I didn't have enough time to shrink resolution. And everyone told me that was fine, but see, it's not fine, Rob. Yes, you're going to get these amazing figures. I'll let you know when they're like 300 megabytes each. You can zoom in forever. Okay, so yes, they will be there in a second. Obviously, the slides will be there to have, so you don't have to make notes. And I think it was already mentioned, but this is recorded and posted online, so it's there forever, which is super scary to me. I've never Googled myself there to YouTube. That would be very scary. I don't want to hear that. Okay, so let's get right into it. So the learning objectives for this module is really sort of understanding the general approaches for characterizing the microbiome and just touching really the difference between 16S and metagenomics, and we'll briefly mention metatranscriptomics and a few others. Then we're going to talk about if we do metagenomics, how, if we're doing sort of host-associated sequencing, how that comes into play with the amount of sequencing depth we need and how that can be removed and challenges with that. And then the main target for this is really going to be how do we taxonomically identify the different taxa, the microbes in your metagenomic data. And so this would be equivalent almost a little bit to if you had 16S data and making, you know, a list of taxa, but using metagenomic data, and we'll see some of the challenges and approaches within that. And yes, so that's basically that. Okay, so let's make sure we're all on the same page here. Methods for studying the microbiome. We are going to be primarily focused almost entirely on metagenomic data within this advanced workshop. And just to make sure we're all clear on that, that's really where we're sequencing all of the DNA in a sample, right? And so this DNA is arriving from usually a mixed culture, either sort of in a natural habitat where it's really just, say, the purest form, like it could be, say, soil in its natural form, or maybe it's even like metagenomic from like a culture enrichment where sometimes people are growing up several microbes together and then sequencing it. Metagenomes is at its heart just genomics, but applied to many different taxa within your sample, okay? The nice advantage of that is that in theory, we get all the DNA, which means we can, in theory, identify all the microbes that are within that sample, right? So we've talked a lot about 16S in the past, and that's bacteria specific, or you can do other amplicons to target whatever you're interested in. With metagenomics, you're going to get all types of reads, and that means in theory, we can identify bacteria, we can identify fungi, we can identify microbial eukaryotes and viruses. And depending on the ratio, obviously within that sample, you know, and how much we sequence, it'll vary in our ability to identify all those things. Besides taxonomic annotation, which is this module's focus, we'll then get into, you know, what are they sort of doing, right? So we get into what genes are encoded within those taxa, how those genes are annotated, how they fit into pathways, the metabolites in theory that they could be producing, things like that. But it is, remember, at the DNA level, which means we're studying genes. As alluded to by John already, there are other technologies as well. So metatranscriptomics is obviously then sequencing all the RNA in a sample. And the nice thing about that is that you can use it again to identify taxa that are within there, but obviously usually the focus is identifying transcripts for the purpose of seeing more activity at that level. There's some other nice benefits in that RNA is turning over quite rapidly, so you're getting active transcripts, meaning that those are coming from alive cells, which is sometimes a little bit of a problem with metagenomic data. They sort of, you know, the classic question is, what are they doing differently now? And obviously that resolution can change quite rapidly with transcriptomics, meaning if you're sampling over time, you can identify those shifts in response to some stimuli. Do remember, though, if you are doing metatranscriptomics, that RNA is much more rapidly degraded than DNA, and so that is a challenge in itself. Whereas with DNA, yes, you looked at me very.
So the slides are actually too large. The website will not preview them, so I'm uploading them to the Slack right now. I will fix that. They will be on the website eventually, but for now, you're just gonna have to download them from the Slack.
I was told twice this morning, size didn't matter. All right. It's all good. So yeah, so RNA turns over quickly. It just means that when you're thinking about your sample collection, right, that you have to be worried more about the stability of the RNA. I don't know if John's usually our metatranscriptomics guy, as you've already heard, and I don't know if, John, are you going to talk more about metatranscriptomics at any point?
I am.
Perfect. Then I will leave that to you. I won't steal all of your thunder at all. There's also obviously other ways to study microbiomes that are non-sequencing approaches, but we see this increasingly used sometimes in combinations and projects. So we can think about obviously then studying at the protein level through metaproteomic studies. And so also on top of that is metabolomic studies, where we're studying the metabolites within that environment as well. And obviously the holy grail, I think, is a bit where we start to combine all these omic technologies to really understand functionally what's going on within a particular environment. Yes, okay, so that's great. Okay, so that's kind of where we're at from different technologies. And now we're just going to get into the challenge at hand with metagenomic data. So if we have metagenomic data, and we have reads coming from across multiple genomes, how do we essentially get a classic list of different taxa in a nice table format, which we can then convert into, say, stacked bar charts, or we can convert into alpha diversity measurements or beta diversity measurements. There's quite a few challenges that's unique maybe to metagenomic data compared to, say, a more basic amplicon-based approach. And so we have to remember that because we're sampling all the DNA in a sample, the reads are randomly assorted, right? In fact, we're hoping that we're getting a nice even representation and that we're not, enriching for certain things or missing parts of the DNA. Usually the reads are short. I will talk a little bit about long reads towards the end, but for right now, if we sort of think about reads, often they're in that 100 to 150 base pair length. That being said, with NextSeq and some of the HiSeqs offering 250 base pairs, we see a little bit of an increase there from Illumina technology. And of course, we do have long read technologies pushing that boundary more. But still, we have a limited amount of information gained in that single read. And that usually, even though increasing sequencing depth occurs as well, and maybe if you have a huge budget, you can sequence a lot, we're not saturating our sampling, right? We have often hundreds or thousands of different species within a sample. Each of those has lots of cells represented within that environment, and they each have a genome, right? And so that means that we're not going to be able to sequence every single cell and every single piece of DNA in that adequately. On top of that, we probably won't even, cover, a single genome very well, maybe for the more dominant things, but considering we often have a rare sort of tail of taxa, those we won't be able to have as many reads. And we'll sort of see how that comes into play a little bit later. Lateral gene transfer should be in the back of your mind all the time, especially for taxonomic annotation, right? So unlike marker gene studies, which in theory use a marker gene that was chosen because it's universal and hopefully not transferred laterally that often, we know that bacteria, microbial eukaryotes, and even viruses will transfer DNA horizontally or laterally. And that means that when we identify, taxa from a piece of recently laterally transferred, all of a sudden it could look like something that it's not. So all these reads that we get may not be great at really telling us what the source organism was for that piece of DNA. There's also some real basic technical limitations as well. So with Amplicon data, we often run it on a server. In theory, you could almost get away with doing some analyses on your laptop. But with metagenomic data, computational time grows quite tremendously, and we need more resources to analyze that data. So, and the primary concern for that is huge databases and also large data sets, right? So we're already sequencing usually 100-fold more per sample. So our query size is much larger. And then our databases get large because we're interested in, trying to map all those reads to different taxa. And that goes into play for function as well. Okay, so if we go right into initial bioinformatic processing steps. So imagine we get our data from our sequencer. I'm not going to talk too much about sequencing here, but essentially, depending on the sequencing technology, often we multiplex samples on the sequencer, and then using that data, we have to demultiplex, meaning that we basically separate it back into the samples that it was barcoded with. So we can do demultiplexing and lane merging. Lane merging is not as big a deal anymore. That was where we'd often have multiple lanes on Illumina technology, and sometimes you could combine those. But essentially in this step, it's often done on the sequencer, but if not, we're basically just putting the reads back into the samples they came from, knowing that they were barcoded. That's straightforward. Quality filtering is quite important here, and I'm not going to talk about it in any sort of detail at all within this. We covered it within the basic, but essentially removing low quality reads is important, and ensuring that, you know, whatever a threshold is going into downstream analyses, right? So same sort of techniques, we're removing possibly reads usually is, we're often not trimming at that stage. We're usually just removing reads that fall below some quality threshold across its length. If we're doing Illumina sequencing, it does allow us to do this paired end sequencing, which I've talked about again in the beginner. I promise not to do too much of this back and forth for those that are just arriving, but it does allow paired end reads which have a forward and reverse read. And with amplicon data, we often merge those so that they overlap and then they're stitched together. With metagenomic data, because the fragment of DNA is more random in its length, and that length is often bigger than the size of the read lengths, then those pairs, the forward and reverse pair, often don't overlap. And because of that, we often don't try to stitch those reads. It does vary depending on your library. If it's well fragmented, then maybe you could stitch them, but usually we don't try to merge those paired end reads. And we essentially often just combine the forward reads and the reverse reads together and treat them as independent. That being said, for the MAG stuff, you will see that the forward and reverse read come into play quite importantly for assembly, because now they tell the assembler that they were on the same piece of DNA. And so it will come into context and used, that information is used. Okay, and then one of the other ideas before you start trying to essentially identify taxa from, say, reads is the removal of unwanted host-associated reads, okay? So if you're working as sort of a purist environmental person with, say, soil or water or some sort of bioreactor, then maybe you don't have to worry about this. But you can imagine if you're talking more about, obviously anything human-associated or animal-associated, but this could also come into play for plants, right? Again, when you sample, however that's being done, you're going to get other DNA that's not microbial related, right? And that could have a significant proportion of the reads in your sample. If it's very microbial rich in your sample collection technique, then maybe this isn't so much a worry. It could be that, you know, 90% is microbial. You're still going to want to try to remove those host reads. You can imagine in other scenarios, that's actually, you can have a significant amount of host-associated DNA, and you definitely have to try to remove that and work with the downstream microbial reads. For all these techniques, usually the standard is to, yes, John.
I was wondering if you were interested in commenting on the ethics of human reads and when you're depositing these data sets to public repositories.
Yeah.
Where we are with that.
Where are we? So yeah, so I guess besides the practical side of bioinformatics and the importance of removing reads, as John alludes to with human samples, it's often thought that usually if we think about ethics and microbes, there's sort of a threshold of, okay, the bacteria that live in and on you is not as much of a privacy concern as obviously your human genome, right? And so you can imagine that if you don't do a good job of removing contaminant reads, then now you're putting, you know, fragments of the person's human genome in a public database, presumably downstream. And usually this is, if the ethics board is well knowledgeable in this area, they'll sort of ask you what you're going to do. And then one big problem though is that no matter what, I guess there's always a chance of some human reads slipping through and going into databases. Besides that, I don't have any strong comments on that. Do you have any comments on that, John?
Really being adequately addressed in terms of when it's being state of the art in terms of what we do in terms of removal. And I think you're absolutely right. The ethics boards don't seem to be quite aware of this potential issue. And when we raised it at SITKIS, they were suggesting, well, you know, you try and be as proactive as possible. And does anybody have any experience of that here? In terms of removing human reads from an ethics, REB kind of perspective? Yeah.
Yeah, so I did like this stuff in the Lightman lab, and there were some papers that had come out of that lab before I joined, I think 2015, 2016, where they were going into databases and they were getting some of the human genomics data and overlaying microbial genomics with human genomics. So I was surprised to, I mean, it makes sense now, the idea of maybe you want to remove human reads as a privacy concern, but I hadn't really thought about it. I was like, oh, that's something else that you can do with these data sets. So it makes sense to me that you would want to remove these things from maybe a privacy perspective, but I hadn't really thought about it before, so it's interesting. I also work in it, so I really thought this thought.
So I think our general practice now is anytime we receive sequence data in-house, then we screen it for the human reads before we then put it on something like a public compute cluster and do all the processing there. But when we're doing it in-house, we're behind the firewall and make sure that all the privacy concerns are taken care of. But it is interesting that there seems to be a lack of direction from the community from ethics in terms of what we really should be doing to that. It is.
I mean, like when we're looking at it, so for example, when you do taxonomic classification of correct and after, you could still have like, if it's a really high host DNA environment, you still have at least 50% of your reads human. They might just be kind of crappy DNA, but that will get rid of it all the time.
Yeah, and there's a question or comment here too.
Yeah, I just wondered, like, very quickly, what happens if you don't remove them? And then I guess my sub question is, in a fecal sample, a lot of the DNA will be from diet, like the plants, and we're never removing them, just sort of like them. So
Yeah.
what are the
Yeah, okay, so there's lots in there actually. So sorry, what was the first one about the non-diet? What was
if you don't the host?
Yeah, okay, so if you don't remove the host associated microbial reads, in theory, a perfect annotation pipeline would just, you know, sort of ignore those, right? And we'll see. For sure that if you give it human reads, and even if you have a database of microbes and the human genome, it'll still often annotate sometimes human reads as microbial, right? And there's reasons for that besides just it being crappy or something. It's, you know, you can think about there's just some similarity between a particular fragment of DNA in the host genome and these microbes, right? But obviously that's false signal that you would not want. So the idea here is that if we're aiming to profile the microbes, it's best to try to remove them as a first step. That being said, obviously, which was kind of alluded to here as well, is that when we say remove and throw away, well, obviously, depending on how much human DNA you get or host, you know, you may be able to use those reads for something quite interesting. So, you know, I had a study with IBD where we actually genotyped the host for a few particular SNPs associated with IBD. You can imagine if you're working in animals or plants would be a little rough, but maybe there's something on the host side you could actually use that DNA for, depending on the ratio of host to bacteria. And then your last point about diet in humans is good, and this would apply to animals as well, is that I think it's been generally sort of ignored until more recently. So it's very hard to say remove those reads for the diet because we have obviously a lot of genomes and diet components, right? I guess we cross our fingers a little bit and hope that those reads aren't associated with microbes, but I'm sure it does filter into some of that false signal. And then actually recently there was a paper that actually took that data and then tried to actually provide diet information based on those reads that did map to food products. And so there's is that Sean Gibbons? Thank you. Yeah, I'm not gonna. Yeah, of course. The Sean Gibbons lab, right? Yeah, Sean Gibbons lab, where they basically, you know, push this idea that from a metagenomic sample, you could actually get the person's diet history. You push the envelope a little bit saying, you know, maybe this is an alternative to food frequency questionnaire questions. We recently discussed this in a DAO mug. I think it's a bit detailed here if you want to chat about it after. I think could be a time and place for it, but obviously there's some limitations there too. Yep, question here.
You don't have a reference genome for your host,
Yeah.
like, there's not even an entire family.
So I guess if you don't have your exact reference host, people would suggest, well, like, what's the closest host you have, right? Because there's going to be some often, sequence similarity, right? If you think about even like mammals or things, right? Then we can think about trying to use the most closest reference genome and hopefully using that. If it's something really unique or unique like some plant that has a very large genome that hasn't been sequenced, then we are limited a bit with that. And I think you have to sort of keep that in consideration down the line, that you might be getting some false signal.
What if it's a really complex contamination, like I say, food waste?
Yeah, so food waste would be really similar to the diet stuff, right? Where essentially, if you can build a database with whatever you want in it. So in theory, if you want to get really elaborate, maybe you start adding reference genomes, as much as you want at the first to try to help filter some of these reads out. But food waste is going to be super, like, that'd be really hard, right? So that's
Undefined, you don't even know what's in there.
no, exactly. So it'd be really similar to the sort of diet situation where we have food products there, but they're going to get sequenced. Yeah, I don't have a great solution there. Yeah, it's a good question though. Yeah.
So like when I see when you think of pipelines, they always like have using the contaminants like host to DNA as a standard. But what if you deal with a lot of the data, you just skip it and or just that.
Skip what type of data?
If these host data,
If you don't have host data?
like just like soy data, like
Yeah. You're good. You're good to go. You just I mean, you might want to think for sure though, is there no other DNA in there, right? Right? I mean, I don't know, like yeah, if it's a pure soil sample, sure. I mean, is there really no plants around though? I don't know. I mean, yeah. It's probably a ratio thing for sure too. Yeah, but obviously if there's no host, then yeah, this is something you can sort of just skip. Yeah. Yeah, Steve.
I like how the ethics started this. I wanted to make a point and maybe ask a question to everybody is, when we use the word contaminant, what do we mean? Like, what is a contaminant in our sequencing data? Is it something in the kit we use? That's probably a contaminant, right? Because that's not from the biological source sample. Is it something we introduce inadvertently during processing in the laboratory environment? Is it something the sequencing center introduces, again, airborne or liquid-borne? Those are probably true contaminants because they're not part of the source. But when we use the word contaminant to talk about a source sample, is that the appropriate terminology? Maybe off target if we're looking for specific types of microorganisms or specific taxonomic groups or domains of life, but really the sample is the sample. It's a biological entity that you've sampled. It could be soil, it could be water, it could be human. And I'm wondering how people feel about that because, Tony studies this, like in the literature, you go into cyanobacterial genomes. And people often exclude the co-occurring microorganisms, the reeds that live in co-culture with the cyanobacteria. They call them contaminants, but they're not contaminants. They live with the cyanobacteria. And in the database, you don't know because they've excluded the data. So it's almost a philosophical dilemma about reproducibility in science. Like, is it better to have all of the information and then to quantify the loss of that information as a function of your interpretive process versus just filter it out, call it contamination?
Yeah, no, it's very fair. Yeah. I think that's why I have contaminants in quotes here, but yeah, because from a technical side, I'm treating like say the phi-X here as often a sequencing control that's usually filtered out by the sequencer. Sometimes it slips through, so we include that in this screen essentially. So sorry to come back to you. I guess you would have to do that step. It's good to have a screen at least to make sure some of these things may not have still be in your sample, even though there's no host associated. And it also comes back a little bit that contaminants like Phi-X, which is technically put in for sequencing control, or if it's a soil sample and it's collected by people that aren't super, worried about introducing their own bacteria, maybe you still want to screen it against human genome because you don't obviously, there's a slight chance there. Usually it's not a practical worry because the amount of bacteria in soil compared to some human sloughing off is usually minimal, but still, why not screen it anyway, right? And then, yes, definitely, we're sort of tossing it here, but you should really think about, hey, could I use those reads? Could I do them something else is with this host associate? And I think in that issue, yeah, it's not really contaminant. I like the term used. What did you say? Off target or?
Off target information.
Yeah, absolutely. Yes.
Yeah, so I had a question. So when you're finished your project and you're thinking then of uploading your raw sequencing data to like GenBank or putting things on SRA and CBI or whatever, are there specific standards for metagenomic data that, because we were talking about like, should you be removing host genes, should you be removing Phi-X, et cetera? Is there somewhere there's like a standardized set of rules, like this is what raw data means in this whole genome sequencing context versus like, if you're doing 16S data, you usually just straight upload fast.
Yes. I don't think there's a standard. I think it comes down to probably trying to describe exactly what you put up, because this happens a lot. Even obviously it comes into play a lot with human samples, but you can imagine other samples. So, just from what I've seen, you see everything from, here's the raw off the sequencer files to here's stuff we removed, possibly host associated. And then also like we did a whole bunch of quality filtering and then just uploaded sort of our quality filtered reads. And so I don't know if there's a standard, but yeah, Ryan.
Well, I was going to say that there isn't, but there are groups that are trying to help standardize it. I'll be talking about that this afternoon, specifically about the data products producer on it. So assemblies, mags, other types of things, filtered raw reads, things like that. There are standards that are being suggested, I guess, protocols to follow, but you have to kind of just know that you should be good with your data and just follow those practices. But yeah, we'll talk about that. So.
Yeah, definitely. If you go in the databases, no, it's the Wild West in there. It's your responsibility to do better.
Consent, people have been. Like how people have consented for their samples to be used in chat.
I think that's all about you do human consent, so I guess that's it for sequencing data, I guess it's I think it's not so
Probably, yeah, I mean, I haven't done a whole of it, I mean, it's a whole ethics mess consideration, but yeah.
We've come full circle back to ethics. I like that. Yeah. But it is a good point. If you're doing human sampling, it's good to think about that data at first, because unlike other things, you can't go back, right? So you have to consent. And I've been in this with lots of collaborations. We're like, okay, we're ready to put it up in the repository. And then you find out that they originally didn't even consent them to do a microbiome analysis. And it's not clear whether we can upload data or not, right? Okay, this is amazing. That was great. Thank you, John, for starting that off. So I did want to sort of, sometimes this comes up as a topic is, hey, if we have a lot of host or off-target DNA, and we aren't interested in it, is there ways to boost the amount of microbial reads like before the sequencing, right? To try to improve that situation. Because the reality is, if you take, say, a stool sample, is the classic one, then most of your reads, 99% are gonna be bacterial or microbial related and not host, right? If you want to do metagenomics of, say, saliva, now about 90% of that DNA, maybe 80% is going to be host associated, right? And now all of a sudden, throwing away 80% of your data is not very satisfying, and it sort of limits then your ability to do metagenomics in particular sampling, right? And that's where actually amplicon base still has a lot of hold, right? Because you just doing metagenomics is not feasible because how many reads are going to be host associated. Of course, you can just sequence more and more, and people have done that. But there is groups trying to come up with, essentially on the wet lab side, we're not going to talk a ton about this, about techniques to try to filter out the host reads, say human, I'm going to focus on here primarily, entirely. compared to the bacterial reads. And this was a paper that came out just last year. And so I just wanted to briefly show sort of what that would look like. And the whole idea, again, is they were using lung samples. And then this is showing, okay, if you do some sort of host depletion, you can actually get more reads, and that will result in more species identification. It would improve your ability to do MAGs and functional annotation, basically everything. In this, they compare different types of approaches, wet lab approaches. I'm not going to get into them. The basics behind pretty much all of them is just leveraging the fact that human cells, when they're extracted, are just much more sensitive. They're disrupted earlier, and bacterial cells are a bit more robust. And there's ways to essentially remove free-floating DNA. And so you can remove free-floating DNA and also try to essentially extract initially the human cells, remove that DNA, and now you can do an extraction with primarily more microbial cells and sequence them. That's the general approach. So this compared different methods. I'm not going to go into great detail about this. I just want to show that these exist. And what they show is here is that this is the ratio of host to bacterial reads for the lavage samples, nasal samples, and sputum samples, and then across the different methods. And they see that you definitely get an improvement, but it varies by the sample itself and also by the method. So that's good to know. And then doing that results in obviously your ability to identify more species. So that's not surprising. So because sputum improved this ratio quite a bit, you see identification of more species. Same with nasal samples. And then, of course, you have to keep in the back of your mind, though, that if you do that enrichment, you could sort of bias away possibly from what the real underlying microbiome is, because maybe that enrichment is not perfect. And so this is just showing for those different sample types as well, and each of those methods, how far that community is from the baseline, from the real community. So bars to the right are higher dissimilarity, and so those things are more different from the base. The one thing about this paper they didn't do great is that removing host also coincides with removing dead bacterial cells. And so I think that's obscuring a little bit of their signal here. Again, maybe that's a good thing, but their baseline is including dead cells, and so it's not clear between those two. But anyway, it could be an improvement. Just wanted to throw that out there. Any questions about that? Okay, great. Okay, back to bioinformatics here. Okay, so we have our reads, we have them quality filtered, we've removed contaminants or host associated or off-topic target sequences. Now we want to get into, okay, what taxa are there? And so broadly, there's a few different approaches to this, okay? And so I call one approach sort of an all reads approach, which is where you take all the data and you attempt, in theory, to assign taxonomy at least to every single read in that sample. And you can do that by using large reference databases. That is computationally difficult because you're doing now all your query reads against a large sequence database. It's also possibly leads to some inaccuracy because, as I mentioned before, some of those reads may not be good markers for taxa identification. Like it could be that they were horizontally transferred. It could be that they just don't have a match in the database, obviously. They could just be very like repetitive element or just not contain much information. But essentially an all reads approach at least attempts to, out of the box, assign taxonomy to it. The other approach is sort of a marker-based approach where you say ahead of time, here's my markers I'm going to look for within my data. I've predefined those ahead of time, and I'm going to start to search for a subset of those markers within my database. So now my database here isn't everything. It's basically these sets of genes or markers, and I stream my query sequences against it in the hopes of identifying taxa that way. The benefit of this approach is it's faster, and two, as you can imagine then, because we've pre-selected these markers ahead of time, our precision's a bit higher because in theory we've picked good markers, right? So if we just focus about marker-based for a second, what would those markers look like? Well, you could actually do this using anything that you like. You could actually pick a universal gene, the same that we would do with amplicon sequencing. You could actually extract 16S sequences or CPN60 or any other universal gene, and then basically do that workflow and identify taxa in that fashion. We don't often do that because, you know, you do all this nice metagenomic sequencing and then you just pull out one gene. It doesn't seem very satisfying, and you may not get to the taxonomic resolution as another approach. The other way is to use multiple genes at a time, and so people come up with different sets of those. MOTUs is well known. They use 10 universal single copy genes, so imagine 16S plus others. And they use that to, again, identify taxonomy, but even then, just 10 genes, most of your DNA is not going to be annotated, and so that might be restrictive. And then the other approach I'll go in a little bit more detail is Metaflan, which has been through many versions, and they're on Metaflan 4. In the lab today, you're going to use Metaflan 3, mostly just because it's a computationally a bit of a bigger hurdle there for Metaflan 4, but the technology is very similar and the accuracy is kind of similar. Yes, John?
I have a question about these approaches. These marker genes that they're using, are these chosen because they're not known to be involved in natural gene transfers? They use them as criteria for deciding these marker genes, you know?
For the universal single copy genes, yes. I've sort of seen those. I mean, there's still a chance that some of them are universally or horizontally transferred, but they are selected in that approach. Metaflan is no, they don't technically look for horizontal gene transfer, but because, as we'll see, they focus on identifying a marker that's only found within a set of genomes. I think they would argue that maybe that doesn't come into play as much. I think their approach sort of biases away from that, but they don't explicitly try to remove, say, genes that are known to be involved in horizontal gene transfer, per se. Yep. Okay, so one of the tools you will be using is Metaflan in the lab, and the other is going to be Kraken, which I'll mention in a minute. But Metaflan 4, anyway, basically starts with a very large number of bacterial and archaeal genomes. A lot of those are from isolate or single cell sequencing, so sort of more traditional roles. And as more and more metagenomes are produced and their ability to make MAGs, assembled genomes from those genomes end up going back into our reference databases now, and they actually outnumber our sort of more traditional genomes. So they actually leverage all of that data, and they just do a lot of binning into what they call species level genome bins, or SGBs. And so that is a cutoff at 5%. And that's basically to try to reduce, obviously, that large number of genomes to a smaller representative set of genomes. And then within that, they start to identify unique and core marker genes. So essentially, any sort of piece of DNA within that genome that is found across one of these SGBs and is unique to that SGB. Doesn't mean that, you know, in the future, if it was horizontally transferred, that they could possibly find that marker now in a new genome, and that would, obviously within that newer version, would probably then get it removed, but you don't know that ahead of time. Depending on the SGB, there's different numbers of markers, 10 to 200, and they also, within the SGB, they'll sort of use subsets of markers to provide greater resolution. Now that number of marker genes still sounds like a lot, but the reality is if you do that with a sequence similarity tool like Bowtie that is very quick, you can actually do that mapping relatively quick, although it does take more memory now than it used to in the past. But those reads are basically from your data sample are profiled against them using Bowtie. So this is a genome mapper, the same sort of tool you would have used in that host contamination step. And then they're normalized to produce taxonomic profiles. A note here is that the database is limited to bacteria and archaea. So if you're interested in microbial eukaryotes, you're not going to get that information. If you're interested in viruses, you're not going to get any sort of taxonomic profiles from that approach either.
But I don't think anyone would recommend it actually.
Microbial eukaryotes?
I don't know the eukaryotes.
Oh,
Only a few.
oh, okay.
I think in 4 versus, I think that was like even 4. And it does have some viruses, like it can identify some, but I don't know that anyone that's interested in those would ever recommend using it.
They definitely had viruses in 3, and I thought they actually backtracked and removed viruses.
I'm sorry. Maybe it's like that they just have a human genome. So I'm just saying.
Yeah, yeah. Either way, don't do it for viruses or microbial eukaryotes. Yeah. Okay, so that's Metaflan 4 approach. The other approach here is an all reads approach. And so for there's quite a few different methods out there. We'll talk about Kraken and Bracken. There's centrifuge, there's Keiju, there's sour mash. There's always a new sort of approach that essentially tries to increase accuracy while also being fast at the same time, because this is that step that's quite computationally demanding. And so there's quite a trade-off between what's you can do in a timely manner versus the accuracy. And so for computer scientists, that's an attractive problem to try to solve and optimize. And so there's always new methods. Most of these methods use a K-mer approach, which I'll describe in a second, to sort of do that fast sequence matching. It does trade off some sensitivity because of that. And then it tries to combine all of those matches, often using a lowest common ancestor approach. Not always, but it's a common approach for assigning taxonomy once you have many marker sequences. So what are K-mers? It's a very basic idea in sort of computer science and genomics and bioinformatics. And a K-mer is if we think about a bunch of different genomes in our database, we pick out a genome, we look at it as a linear sequence of letters, and then we define a K-mer where that K is a particular number. And so we then break up that sequence into all those short fragments of DNA. So in this case, I'm showing 5 mers, right? So that would be a K-mer of length 5. Depending on the algorithms, you can usually get down to some option where you can actually change that K-mer length. And you can imagine a short K-mer is usually faster, but not as sensitive as a longer K-mer, but you may miss something. Now, this doesn't look like a great way to speed things up. You take one sequence and you combine into a whole bunch of short sequences. But the reality is you can use K-mers to build these nice algorithm approaches of databases. Essentially, they're called often suffix trees. I'm not going to go there, but essentially we can because of the length and the way that they're categorized, we can actually identify those very quickly with particular algorithms. So that's why they're used as an approach. The Kraken, Keiju, sour mash, if they're a K-mer-based approach, they all sort of maybe start with this. They use different databases. They use different algorithms to search those K-mers, and they use different K-mer lengths, okay? Length? No, not at all, really. I think, I don't remember what Kraken uses. I want to say it's like in that 8 range.
31.
Okay, 31. Yeah.
Is it something where you would like try different numbers?
No, not usually. I mean, you could technically, but of all the things you can probably tweak, it's probably not one you're going to go after. But you could technically, yes. Yeah. It is obviously defined when we're building the reference database as well, right? So there's options you can vary when we're doing the matching, as we'll see in a second. But the K-mer size would be restricted to the database building stage. And so it's, usually the makers of the tool are trying to optimize that for you ahead of time.
I think like in the papers, they'll actually demonstrate.
Yeah.
The K, the effect of K.
Yeah. Yeah. I mean, that's the one thing that they will do really well is they've already tried to find the maximum K for you. Yep. Okay, so that's great. And in most of these approaches, you eventually come to a point where you have, some sort of marker or k-mer or multiple k-mers assigned to a reference genome or SPG or species or whatever, however you want to define it in your head. And then you say, okay, well, that's great. How do we then actually spit out what taxonomy it is, right? Like, and at what level? And so you can imagine if the marker that we're looking at is shared across different species, or in this particular case, a check mark means a different genus. The question is, at what should we assign, that read for taxonomy based on this information? And this is just a simple sort of toy example. And so if there was 2 check marks for Escherichiae, I can never pronounce that properly, then we would then obviously assign it at the genus level of that and not know what it is at the species level. In this case, because we actually have 2 different genera, then we would actually not even be able to assign it at that level very well, and we would push it up to the family level. So this is why for many of these methods, sometimes you'll get an identification right down to a species level, right? If all the markers are clearly pointing at just one species or even a strain, we can get that sometimes. But if the read basically has multiple things it's hitting, it'll often get pushed up to a higher rank. And your resultant file, you'll have to think about that, about when, at what taxonomic rank you do your analysis at. Different tools will tend to have lower, or the rankings more higher resolution than others sometimes. In the particular case of Kraken, which is a k-mer-based approach and is the one we'll sort of talk about in more detail, it does this LCA as well on top. And then they have actually combined that with another tool called Bracken, which you essentially run after the Kraken method. And it tries to actually sort of take those initial set of LCA reads and then actually try to get a bit more resolution by pushing them down to, say, the species or strain level. And it does that because essentially you can imagine if you have a database that's growing higher and higher, there's just going to be more shared content, and you may end up getting a higher taxonomic rank, but you know that it's more likely that there's just a single set of species within that. And essentially it'll try to push those reads down so that you have better resolution at that level. I don't think I want to go into more detail than that, but essentially, if you're running Kraken, it's well recommended that you would run Bracken after it to get a better taxonomic assignment. Okay, so with 10 minutes left, where are we at? Yeah, okay, I can pull this off. Those are the taxonomic assignment sort of approaches broadly. And then the big question is, okay, you've come here, you want to know which one should I use, which one is best, the classic. And so this is really difficult to assess. It's between the tools because the tools are constantly coming out. They're using different reference databases. How they measure their accuracy varies. And so it really depends who you ask and who's done the analysis. If you look at one particular paper, absolutely, they're going to show that usually their tool happens to be the best. And then you look at another paper and they've also showed that their tool is the best. And so it varies quite a bit. We were interested in this a few years ago, went down this rabbit hole, and we still are there, unfortunately, about really trying to compare mostly Kraken and Metaflan, because that's 2 of the most popular tools. And yeah, we went down that rabbit hole. So we had this paper came out a couple of years ago, and I'm just going to show a couple of results from it. This slide has a lot of stuff covered up in the middle, so it's not very pretty. But one of the take-home messages I want you to really appreciate about the trade-off between a tool like Metaflan, which is this marker-based approach, and say Kraken, is really there's a vast difference in the number of things you're going to identify. Okay, so this is a human-associated samples, and I know you have to squint a little bit, and this is the number of species identified for all those samples. And this is showing how many things you get from 16S for this particular sample. There's lots of reasons why 16S may not agree with metagenomics. We can talk about that sometime. And then this is showing metaflan 3. So we found in this particular case an average of 64 across the samples compared to 117. Not that big of a deal for whatever, I'm not too worried about that, as we'll see later. And if we compare this to Kraken, the defaults would give you 22,000 plus species, right? I mean, 22,000 versus 100 or 60 is really different. If we increase their confidence cutoff, which is this ability to limit the predictions to things that's more confident in, then we can reduce that number to 568. That's at least in the sort of correct order of magnitude. So then from this, you'd be like, oh, well, maybe I should just use metaflan. And to be honest, if you have human samples, metaflan is going to do a great job. And I think it's universally thought that why not? If you go to environmental samples, non-human samples, samples where we don't have great representative genomes ahead of time to make these markers, then this problem gets a little bit blown out. So now what we have here is the 16S for these set of samples showing 426 species, and metaflan on average is showing 0. So that's quite problematic, right? We know there's more diversity there. So it basically sort of falls apart. This is metaflan 3, by the way. So disclaimer, asterisk note. Sorry, Curtis, if you're looking at this lecture on YouTube right now. we can improve it. And then Kraken obviously now identifies, again, tens of thousands of things with its default cutoffs. And if we apply cutoff of 1, we sort of get it back down to the right magnitude according to 16S. Okay. So this is good to understand that, one tool obviously is not going to work in both situations. And we got quite interested in essentially trying to improve maybe Kraken as a starting point to make it in the right range, because we did try some things with metaflan to improve it. We just couldn't get there. There was more dials to change and more easier reference databases to change with Kraken. So I'll skip over most of the results from the paper. This is just showing 3 different results comparing Kraken 2 with this complete database that we built. This is Kraken 2 with essentially metaflan's chocoflan database. And then this is metaflan on the bottom using 2 different cutoffs. And so this is showing the proportion of reads within your sample being classified across these different tools and databases. And this is that confidence threshold I mentioned earlier. So this is a way to improve, in theory, precision while trading off some recall, meaning we can't identify as many things. With this particular set of samples that we were using, then we did show that we could get, sort of optimize the confidence threshold for Kraken to, maximize this mean F1 score. F1 score is essentially accuracy, right? So a trade-off between precision and recall. And then that we could essentially get a better F1 score than, say, metaflan with this particular cutoffs of in that 0.5, 0.6 range. The default for Kraken is 0, just to let you know. So then based on that, we sort of had, some broad suggestions. And I think last year I was saying, if you had human associated samples, metaflan or metaflan 4 would be a great approach. It's fast, low computational requirements, very out of the box, straightforward to use with good precision overall at the cost of some recall. If you have more environmental samples, or you want to sort of expand outside of bacteria and archaea, or just be able to identify more things in your samples, then you really will have to go to sort of probably a more K-mer-based approach, like Kraken, or there's other approaches out there. But be aware that you're going to have a lot of false positives come out of that as well. One way that we suggested to get around that was increasing this confidence cutoff value to maybe 0.5 as a starting point. And then the classic thing is that was based all in simulations. And we published a paper, Robin published a paper. And then we started to do, moving ahead with some other metagenome studies. And real life samples are always a bit different than even simulations is the reality. And so this is just showing Kraken with an increasing confidence threshold for the number of species identified across stool, seawater, soil, and tumor. And the biggest thing that I guess I'm trying to show you here is that for our environmental samples, again, like soil and seawater, you know, the proportion of reads classified by Kraken drastically drops off with this increased confidence threshold that we just suggested you take, right? So now with 0, at least we're getting maybe 20, I guess it's 30 to 50% of your reads assigned to something, where now, yeah, that's great. We've removed some of our false positives, but we're getting drastically lower number of reads classified. And the reality is, if you're interested in something like that, well, then, you know, you're trying to still maximize what you can annotate in your sample. So again, why not? This is where, yes, John.
I think you skipped over the final bit, which is how do you choose that cutoff then, that confidence score?
Yeah, so yeah, we have a flow chart. But the reality is that I guess the take-home message here is that is you can actually try different cutoffs and then look at your number of reads classified to sort of help satisfy yourself a little bit. You can also then see how much the profiles change over time. But I think if we're talking about sort of really environmental samples, you know, 0 seems bad. 0.1 helps quite a bit already. But once you push it up to this 0.5, you are going to get quite a reduction in the number of things.
But your recall will drop.
Your recall is basically dropping. It's just classic problem of your recall dropping, the number of species you're going to identify.
Definitive. I wouldn't know what is this.
So I guess the recommendations, if you're going to use Kraken, not to use the default 0, to probably use at least 0.1 or something. But maybe you can just use Kraken and then take your identified taxa and try to validate them a different way. So this is our most recent little adventure, is over here. And the idea is you sort of use Kraken as this way to say, okay, all these things are here. How do we start to remove this long tail of falsely identified taxa, essentially, right? One way is that confidence cutoff built into it. But maybe there's another approach too that's even a bit more rigorous and more interpretable, I guess, is the question here. And so this is a little tool that, little pipeline, I guess, that Robin's put together. We both acknowledge this is not crazy bioinformatic pipeline or anything. It's quite straightforward, but I think it's quite useful, more probably on the visualization side. So essentially you take your taxonomy IDs identified by Kraken, or it could be identified by other approaches really in this way. You extract the reads from those FASTQ files. You pull out the genomes just for the things that Kraken was already said there, right? So we're reducing that reference genome space because we're basically saying it's already kind of overestimated that. And then we're just going to map those reads back to those reference genomes and sort of look at how many of those reads map to the reference genomes that Kraken said it should. And 2, like, what does that coverage look like of the genome, right? Do we get a reasonable amount of those reads covering lots of genomes, giving you confidence that that genome's really in the sample? There's lots of different metrics you can also calculate, and we tried different ways to do this, but the reality is that we can do this by looking at it visually and then also a few different metrics. So this is an example from some blood samples, which is notoriously quite low with obviously bacteria, and also has, you know, I would say now quite notorious for false positives within it. And most importantly was, you know, when we were doing this, fun enough, this was also published by others, Mycobacterium tuberculosis was often being identified in a large portion of the blood samples. And Mycobacterium tuberculosis causes tuberculosis. And, you know, out of the gates, we thought that was a false positive. But why would it keep popping up here all the time? So this is just basically showing some of the output from the genome coverage checker. what's shown here is that if it's darker blue, it's less coverage across the genome, and then bright yellow indicates that we have lots of coverage, lots of reads across those points in the genome. On the right here, we have this average sequence identity from Bowtie. And then we have some metrics on the right as well, and we're just showing 3. So the first column is the number of reads that Kraken assigned. So this is number of reads. So in all these, we have sort of thousands to tens of thousands. And then after that, we show the genome fraction covered by Bowtie, and then the fraction of these reads that were possibly mapped by Bowtie to its reference genome. So it's basically saying, hey, Kraken, did you do a good job? We're going to double check you with a mapper to the reference genome. And so we see, you know, for this genome, looks great. I would feel confident that it's there in the sample. Is it still possibly a contaminant introduced? Absolutely. But at least bioinformatically, I can say the DNA was in the sample. I feel confident. Pretty confident, like, you know, as confident as you can be in microbiome analysis. And then with this one, we obviously have less coverage, but the average identity is still quite high. We still have quite a few reads, 28% of them being assigned. And that could be a result of just, you know, the fact that it's more rare. And so we just don't have as much coverage, right? We're not going to have a great coverage for more rare things. But down here for Mycobacterium tuberculosis, we basically get 0 reads mapped. And we started with 68,000 reads, and none of them mapped to the reference genome, right? Why is that? The computer scientist in me would try to like figure that out and, you know, figure out why the algorithm is absolutely falling over. We have not gone down that road. We basically said, hey, it doesn't work. This is not just a Mycobacterium tuberculosis issue. It does this for other samples as well. At least now we can sort of maybe then use some of these metrics or visualizations to reduce our number of taxa to a more confident list. Okay, that good? Wonderful. Okay, so then just a last couple of talking points, and then we're going to go to break. I'm just a little bit over. So far, I was talking about taxonomic annotation on reads, right? Taking the raw reads and annotating each one of them. And then we'll see later that we can assemble reads, right? And Ryan's going to talk about that, what that means. But some people will be like, well, why don't you assemble them first into longer pieces and then annotate those? And we can, right? And you will. We can do that as well. One downfall to that approach is, yes, you gain information, but one downfall is that you can imagine, because of this issue where we have some species assembling well because we have enough sequencing depth, we would then taxonomically identify more abundant things. I mean, that's true anyway, but it does mean that for some of our assemblies, we're not going to be able to assemble the more rare things. It's not wrong to assemble first and then do some taxonomic annotation. It's just, it's a difference of opinion a bit, I think, in that range. Some people will do essentially the taxonomic assignment before assembly, and then usually do assembly in mags and do something else after for the microbes of interest for you. But you'll hear much more about assembly in the next module, so I'll just punt that. The other big question is we've talked about short reads, right? So mostly Illumina, right? With sort of 100 to 200, maybe 300 at the max. What about long reads? Obviously, there's an increase in people doing long read sequencing on metagenomic samples. The cost is continually coming down. That could be PacBio or nanopore sequencing. And so those produce long reads. In theory, that's great, because a longer read now should ensure your ability to say this is identified as this taxa. We overcome a lot of issues when doing that. And then you'll see definitely again in the mags, long reads are really useful in that area. But what would you use for taxonomic identification for long reads? Because the tools I just talked about, you know, could have advantages or disadvantages. I am not going to go into this great detail. I just wanted to show at least this one paper that has done a comparison on long reads across multiple different methods. I thought they've done a pretty good job of trying to figure this out. This is 3 years ago. I'm sure things have changed already since then. If you have nanopore or PacBio data and you're trying to annotating it, I think it's definitely a wide open area right now. But you can at least look at these two plots to kind of help you. So on the left, they're using a mock community. Mock communities are great because you know what's in there. There's 17 things here. The reality is mocks don't always give that best picture because you can have lots of things that are not sequenced and it's not clear how your method will perform on those. But essentially you can see that they basically break this down into precision, recall, and F1, and I don't know what F.5 is. I've never heard that actually, I just saw that. Oh, is it equal weighting of the F1? Is it balanced accuracy? I don't know either. I'll Google it after, find out. Let's ignore that. Precision recall. So again, we see recall very good with Bracken and Kraken here. Typical. Precision not so hot. So that's kind of matching what we see with the short reads. Here's Metaflan showing a better F1 overall out of the defaults. And then they show some other methods that I have not talked about. Sour mash here, this BugSeq, and then also MMSeeks is been mentioned a lot by others. Megan is a tool I don't talk about, might get mentioned in function, but Megan's a tool that's been around for a long time. It's just another approach, and they have some specific long read taxonomic annotation methods, which they highlight here for particular sequencing approaches. So, yeah.
Right, what's the difference between precision and recall?
Yeah, precision. Yeah, I should have probably introduced it considering I've used those words how many times in the past 20 minutes. But precision is your ability to, okay, let me just see if I can define this clearly. So precision technically is the number of true positives over true positives plus false positives. Yeah. In other words, it's your ability to make sure that you correctly identify the thing that you were saying is there. Recall is true positives over true positives plus false negatives. I'm pretty sure. And essentially it's the way to how many of the things do you correctly identify within the sample that you knew was there, right? It is analogous to sensitivity versus specificity if you come from a more clinical world. Yes. So maybe I should have led with that. And F1 essentially is different speak for accuracy, which would be often used in more clinical settings with sensitivity and specificity. Yeah. Okay, and then on the right, sorry, just to finalize this is just showing the ability to the proportion essentially annotated to whatever taxonomic rank. And it's showing that, you know, some of the methods get down to the genus or species level and others do not as good a job and are sort of limited to higher up taxonomic ranks, even in this, you know, 17 species well-defined setting. So there's lots of things to consider, I guess, when you're trying to evaluate a read assignment approach. Okay. Any questions about taxonomic annotation? Yes.
Never mind. The Bracken and Kraken,
Yeah.
why does Bracken have less subspecies level? What is such things to this?
I don't know if it's less. I think it looks identical to me, doesn't it? Oh, you're looking at B, sorry.
Oh, okay. What level you wanted to assess stuff at, so it could just be that they were.
Oh, yeah, this is a full bar species, so they must.
Yeah.
But I don't know why they would, I don't know why they would do that. Yeah, I don't know. I clearly don't know the answer to that one. Okay, any other questions? Okay, that was a great discussion. That was really good. I know we're a little bit over time, but that was fun, especially, you know, that whole contamination discussion was great. Okay, so we are going to go on break.